Welcome to This Explainer. Today, we're setting out on a truly fascinating journey to decode the microscopic architecture of the human body. We're going to uncover the biological role of epigenetics, exploring this hidden layer of instruction that literally shapes you at the molecular level. Okay, let's dive right into this. I want to hook you right away by posing the ultimate cellular paradox. If your DNA is identical, why do your cells look so incredibly different? Think about it. Almost every single cell in your body has the exact same DNA sequence. It's kind of like they all have the exact same instruction manual, and yet they create vastly different physical structures and perform completely different functions. So how is that even possible?
Section 1. The Central Dogma. Information Flow.
To really get to the bottom of this, it's super useful to build a mental model of how information actually flows inside our cells. We can basically think of this as a one-to-one-to-one relationship. Information is stored securely right at step 1, a stretch of DNA called a gene. That information is then transcribed to produce step 2, a semi-transient strand of messenger RNA or mRNA. Finally, that mRNA is translated to put together step 3, a protein. So the information flows in this very specific one-way direction.
Now, what's truly staggering is this number, 20,000. Humans have about 20,000 different genes. Because every single gene holds the potential to create messenger RNA and ultimately proteins, it means this whole process of gene expression is happening constantly, on just a massive scale inside your body right this very second.
Let's look a little closer at the first jump in our model, going from DNA to mRNA. We call this transcription. First up, we have our stable DNA. That's a double-stranded nucleic acid that uses the base thymine, represented by the letter T. During transcription, an enzyme called RNA polymerase binds to the DNA and acts, well, almost like a zipper. It unzips and reads the strand to string together a complementary copy. And that copy is the messenger RNA. Notice how it's a bit different, right? It's single-stranded, and it swaps out that T base for uracil, or U. It effectively copies the information sequence from the DNA so it can be safely carried out into the cell.
Once that messenger RNA is created, it attaches to a ribosome for the next phase, which is translation. The messenger RNA sequence is read in triplets, known as codons. Each specific three-letter triplet dictates exactly which amino acid is needed. So the cell grabs these amino acids one by one and strings them together into a long chain, physically translating that genetic code.
Those long chains of amino acids then fold into proteins, and honestly, these do the actual heavy lifting, the real work of the cell. They catalyze metabolic reactions, provide structural support, and handle cellular signaling. But I really want to draw your attention to that very last point, gene regulation. You see, some proteins are actually transcription factors. A great example of this is the p53 tumor suppressor protein. These specific proteins can loop back, bind directly to the DNA, and regulate the expression of other genes, making it easier or harder for them to be copied. It's a brilliant feedback loop.
Section 2. The Cellular Paradox. Identical Sequences.
Now that we've mapped that out, I want to bring us back to this stark biological reality. Identical instructions, completely different results. Outside of a few rare exceptions like gametes or certain blood cells, every single cell is working with the exact same sequence of A, T, C, and G letters. Your cells are completely genetically identical, and yet they are functionally entirely different.
Section 3. Enter epigenetics. Gene regulation.
So let's define epigenetics clearly. Epigenetics refers to gene regulation based entirely on the physical 3D structure of DNA. Now, you'll often hear people use confusing sheet music metaphors here. They might say, the DNA is the sheet music and epigenetics is how the musician plays it. Let's just avoid those metaphors entirely, as they usually just muddy the waters. We're going to focus purely on the actual physical reality happening at the microscopic molecular level.
In complex organisms like us, DNA isn't just some straight string floating aimlessly in space. It is incredibly organized. We have 23 pairs of chromosomes, which represent a massive, incredibly complex 3D structure. To organize all this information, our DNA is physically wrapped and coiled around specialized proteins called histones, forming these elaborate 3D structural coils.
And this brilliantly illustrates exactly how epigenetics regulates your genes. When DNA is tightly wrapped up, that RNA polymerase enzyme literally cannot physically access it. Those genes are effectively hidden and silenced. They simply will not be transcribed into mRNA. But look at what happens when the DNA is loosely wrapped. Those sections remain open and accessible. The The cell can easily read them, express them, and build the corresponding proteins. It's all about physical access.
Section 4. Cell differentiation. Fate determination. We can actually trace the whole journey of our cellular development right here. Starting at step 1, we begin with embryonic stem cells. Now, these are generalized cells that have absolutely no specifically defined role in the body yet. As development continues to step 2, they undergo a process called cell fate determination. And while the exact starting mechanisms of this are still a bit of a mystery, it leads us directly to step 3. The cells eventually take on completely unique epigenetic regulatory profiles.
So, the crucial point is this ultimate reveal. Liver cells, hair cells, brain cells, and skin cells all share the exact same sequence of DNA letters. But they look and act entirely differently purely because of which genes their 3D epigenetic structure leaves loosely open for expression and which genes it tightly wraps up and silences. A liver cell is a liver cell because its physical epigenetic profile leaves the liver genes accessible, while the brain genes are wrapped up tight and completely silenced. It's absolutely mind-blowing.
Which leaves us with this final, truly provocative thought. If your 3D epigenetic structure determines every single cell's fate, what happens to your body when that microscopic architecture breaks down? If those complex regulatory networks go awry and the wrong coils loosen up while others tighten up incorrectly, this breakdown in regulation is directly tied to diseases like cancer. Our entire biology relies on this incredibly delicate, microscopic origami. I really hope this explainer has opened your eyes to the fascinating world of epigenetics and leaves you hungry to learn more about the incredible machinery operating inside you right now.